Well, I think you might never guess where I found those microorganisms that you can now see behind me. I found them, I'm gonna just tell you right now, I found them in a chess board. You know, the board game chess? Well, I found them in there. Yes, that's the one. It was part of a public swimming pool, and you might now wonder, where in the world are you going to find those critters here? Well, I will tell you at the end of the video, but for right now, well, have a look. Where could they be? Yeah, stay tuned. I'm going to tell you later. Well, first of all, hello and welcome. Microbehunter here, microbe hunting on the chessboard. But before I collect those microorganisms, let's play a little round of chess here. These are gigantic chess figures made of plastic, and I isolated the sample there and I found this interesting looking biofilm. So it is a very dense mesh of microorganisms and algae and bacteria, and right now we look at them under the low power magnification, but then when you actually go up with the power a little bit, you can already start to see a little bit of movement there, and the individual microorganisms and mostly ciliates and bacteria moving around here. Well, I consider this quite fascinating that even a chessboard is able to be the habitat of so many different life forms. So I spend quite a bit of time observing those microorganisms under the microscope. Well, over this here, what we see, this seems to be a fungus, and those round structures that you see, these might actually be the spores of the fungus, and also quite well visible is the filamentous nature and network and everything. It's quite fascinating. As a focus through the whole sample, different parts of the specimen go in and out of focus, and then again I'm able to see those moving ciliates. If you look very careful, then you can also see that they actually have tiny hair. Yeah, this here, seems to be a so-called conidium, and conidia are the spores of a fungus as well. Yeah, here that's the chessboard again. Now, where in the world here did I find these specimens and these microorganisms in this water sample? As a matter of fact, a little bit of a surprise myself, but it shows how resilient life is, and how many different places you can find it. I took the water sample, I placed the biofilm that I isolated on a microscope slide. I had to separate it very carefully a little bit, not agitate it too much because I did not want to destroy the structure too much. Cover glass goes on top, of course, and then everything has to go under the microscope, and that's basically where I found all of those creatures. I think it's quite fascinating to see always different types of microorganisms. Yeah, this, for example, seems to be the pollen of a flower, because it has this typical triangular shape. I'm not quite sure about this, but it's at least many pollen have these shapes, and also the size was approximately correct. Yeah, and then again in the center here, surrounded by a network of bacteria and algae, you see again those ciliates, and I'm going to zoom in now a little bit more, and then you can actually start to see those hair on the surface, and as they're moving, these are called cilia, and those microorganisms use those cilia not only for movement but also for obtaining food. And then now we go in a little bit further, and then those long small structures that you see, these could also be bacteria. Also shows a little bit the size difference here. Yeah, I think quite fascinating what a chessboard is able to reveal. But then again, I was a little bit uneasy because the biodiversity was not quite as high as I expected. So there were not so many different types of species that I was able to find. So if you go to other sources in the environment, pond water for example, you find sometimes many more different and also maybe even more interesting microorganisms. But then again, I was quite happy to have found those. Yeah, I'm now adding a drop of methylene blue to stain everything, and this actually makes the algae and the bacteria and the microorganisms take on this blue color. And the further away I move, you can actually see the front of the stain of the color moving along, and further inwards the stain was not able to reach those cells, and therefore they did not stain very blue. Methylene blue is a very common stain, and it's quite popular. It stains DNA and increases contrast quite a bit, but it is unfortunately also able to kill some water organisms, so you have to be a little bit careful not using too much of the stain. But I think it also increases the contrast quite a bit, and it's quite interesting to see how the different parts of the specimen take on the stain to a different degree. Yeah, so that's basically how I've been experimenting around, and I know you're still waiting for the answer to the riddle. Where in the world did I get those specimens from? And I'm actually gonna tell you right now, just in a second. As a matter of fact, the figures, the plastic figures that were used to play the chess were quite heavy, as I already told you. And yes, they are filled with water to kind of weigh them down a little bit so that they are a little more stable. And there was the biofilm, because as you move those chess figures around, there was the water splashing around, and this actually created a very moist and wet environment. And over the weeks, over the months, this biofilm started to form. I also tried to look at the biofilm of a black figure, plastic figure, but of course it was more difficult to see, and I can imagine that the black color also absorbs more of the sunlight and therefore also generates more heat, and this probably could harm the growth of the microorganisms. So I took a little syringe, I tried to scratch off some of this biofilm, and yeah, it was not quite so easy to do that, so then I decided to pour out the water a little bit. At this time I also would like to thank my patrons and my GoFundMe supporters, of course, because without you this channel would not be quite as successful as it is right now. In the description below there are a whole bunch of links that I would like to invite you to have a look at, and I think there are quite a lot of other interesting projects that you might be interested in. Well, yeah, that's basically all I wanted to say today. I hope that you enjoyed it. Please do consider subscribing, and yeah, I think microscopy is quite interesting because there's so many different places where you can find interesting microorganisms and specimens. I wish you all the best. Happy microbe hunting as always, and see you around next time. Bye bye.